Thank you. Okay, thank you for kind introduction, Victoria. Good afternoon, everyone. Can you see my title slide again? Okay, good. Actually, I will talk about all the same with different names. It's all related to antibiotics and bacteria this way or another. And today, it's an honor to present on topic of my professional interest, and we will talk about translation accuracy. And it is also a challenge for me, as I realized, because this is probably the first time that I present my work to the audience of such a wide professional interest. And I want to be accurate in description of translation accuracy, but I also want to be understandable. That's why I will take some time to give you like an overview of biological problem that we will be focusing on. So translation is, in plain words, is a synthesis of proteins in our cells. So proteins are the workhorse of life. So if we think of life, we think of proteins. So this would be an introduction. Then we will go down for some more specific topics, and then we will go down to my humble contribution to this field. When we think of life, first what comes to my mind is, of course, a cell. So we think of cellular life. And there is a famous quote of German doctor and scientist Rudolf Virchow, who proclaimed that each cell comes from another cell. So we don't talk about first cell. It's a different story. But it's actually a very deep principle. And I took liberty to show you how it actually looks like. It's never boring for me, although I saw it many times. So you actually see the cell and it divides. And then it divides again and again. And for you, it looks like just some probably exciting video, and you see some green specks, red objects moving on the screen. But actually what you see is all enabled by proteins. So if we don't have proteins, we don't have life. And that's why it's very important to understand how the cell produces proteins. And actually, when we look at the cell, so this is actually a photo of a single-celled intracellular parasite, Mycoplasma. Mycoplasma is kind of dangerous. It causes several diseases. But here is the model of cell to the right. And you see small yellow and blue objects. These are protein complexes that help this cell move, adhere to our cells, infect our cells, and so on and so forth. And yellow objects, actually, it's not a single protein, it's a complex of many proteins. It's called ribosome. And ribosome is actually a factory for protein synthesis, I would say. So this is the main topic of my talk. How this factory works, how we can manipulate it to our benefit or our harm. Let's begin from what we call a dogma of molecular biology. So information in a cell comes from DNA. DNA is like an instruction inside of our cell. And to make protein is like to make or bake a cake on the kitchen. You need an instruction, you need a recipe, you need an ingredients, and you need a kitchen. And then you make a cake. And so the recipe is the DNA. But you don't bring all your book of recipes to the kitchen. You can just write on a page just a single recipe you like, and you go to the kitchen. That's how DNA works too. It makes a single instruction for a single protein. It's called messenger RNA. So the mRNA goes to the protein factory, which is ribosome, and the protein factory makes a specific protein, just one that cell needs at that moment in that cell. So if we go deeper into protein synthesis, so you can see here this brown object is a ribosome, so it's a protein synthesis factory. And this protein factory needs a recipe. This is our messenger RNA, but it also needs the logistics. So to make something, you need a components. Like if you need to build a house, you need the bricks. So these bricks are brought by another kind of nucleic acid called transfer RNA. And these bricks are amino acid residues. So factory accepts these bricks one by one and makes a long chain. And this chain then gets folded into a unique shape, and folded protein chain causes an enzyme. And enzyme does all the job in our cells. Let's just go over from a different angle. So we have a factory, the ribosome. We have a recipe, the red string of nucleotides. And we have a small logistic partners that bring building blocks to make a protein. And that protein folds into a unique shape, and then it works in our cells. It controls all the vital functions in our organism. Here are the names that I will use again and again in my talk. So we have a ribosome. tRNA is a small molecule that brings amino acids into the ribosome. mRNA is our recipe to make certain protein, and each protein has its own recipe, of course. And so the key process over here is a recognition between what's written in the recipe and what is brought by the tRNA. And this is all enabled by a chemical bond. There is an establishment of complementary bonds between letter A and U. These are complex organic molecules. So there is so-called complementarity between A and U, G and C. When it's established in triplets, so there should be 3 bonds in a row. So it's correct amino acid. That's how factory recognizes that the recipe is read correctly. And this correspondence between a codon on the recipe and the anticodon on the tRNA is described or is known as a genetic code. So the genetic code is misinterpreted or misused in a culture, in a media. There is no such as a genetic code of Ukrainians or any nation. It's just a table of correspondence between nucleotides and the amino acids during translation. But of course, it has many meanings beyond the molecular biology. So here again, we have this molecular dogma that information flows from DNA to protein. So the DNA keeps the instruction, and it's very important for cell to keep instruction clean and correct all the time. That's why cells invest a lot into accuracy of DNA replication. So DNA reproduces itself, well, with the help of proteins, of course. And there is a really high accuracy of replication. Then there is a transcription process of rewriting one small piece of DNA into RNA to make one specific protein. And as you can see, the error rate of each process goes down. So cell invests a lot into very accurate replication. That's understandable. You want to keep your recipe book really accurate because it's like a master book for all the proteins. Then transcription is less accurate. And then translation is 1-2 orders less accurate, but still it's amazingly accurate even at that level. It just means that replication is exquisitely accurate. And why is it so? Well, for evolutionary principle, as I said, we just keep our main book of recipes really accurate, and all the rest are just transient. They come and go, and instruction stays the same, or almost the same. Okay, so for a long time, it was supposed that protein synthesis machinery, the ribosome, is like a passive player. It doesn't really care about what it translates, how fast it translates. So it does it uniformly, and it's blind to the type of mRNA and all the situation in the cell. That situation became to change around 1960s. There was interesting, I would say, a groundbreaking work of Professor Luigi Gorini, who showed some peculiar fact. You can make a bacterial cell dependent on certain amino acids. It's like that bacterium cannot grow without amino acid called arginine. So you need to add to the food if you feed the bacteria under certain conditions, and the food should contain arginine, otherwise cell will die. But interestingly, Gorini discovered that if he added a tiny amount of antibiotic called streptomycin is used to treat tuberculosis. So you add a little bit, not enough to kill bacterium, but enough to cause something which helps that bacterium grow in the absence of arginine. That was kind of puzzling, and many researchers probably saw it, but they just dismissed that fact. Like, it's something, some artifact, some incorrect work, something not related to real observation. But Guerini was, I would say, persistent enough to study that mechanism, and actually he discovered the mechanism that controls the accuracy of protein synthesis, the accuracy of translation. So basically we have our mRNA, our recipe for a certain protein, and we have these tRNAs lined up along the mRNA. What we have over here, we have here the arginine codon, and we have uncharged tRNA. So because we have no arginine in cells, there is no way to produce tRNA loaded with arginine, and that would serve as a stop. Cell would stop at that moment, and it would die. But somehow, when you add streptomycin, it lives on. And he discovered why. He discovered that actually cell becomes relaxed in its accuracy of protein synthesis. So let me show you in the next slide. So basically, there is a physiological norm. Let's say you have this triplet AGA, triplet codon AGA, and it is decoded only by this small molecule because it has complementary triplet UCU. This how would happen. All the other tRNAs would be rejected by the ribosome. It would say it's not what they need to do. But if we treat cells with streptomycin, or if we mutate a single protein within the ribosome, so the ribosome itself is a protein complex, a huge protein complex, cell becomes indifferent to what it includes into protein. It could include arginine, it could include another amino acid, and it would produce wrong protein, maybe even toxic protein, but it would do that. So we have so-called ribosome ambiguity or relaxed ribosomes or erroneous ribosomes. But we also could have another scenario. Let's say we could have a mutation which would make ribosome very, very accurate. So much more accurate than the physiological norm. And then it would reject all the ribosomes. The ribosome would reject all the tRNA loaded with amino acid, both arginine carrying and lysine carrying. It would accept them, but at much slower rate. So the speed of protein synthesis would be really slow or even stagnant. This was kind of very unorthodox at that moment, because no one thought that ribosome could be more accurate than it is in nature. But now we know that actually the physiological level of accuracy is not a maximal one. So we have here certain level of accuracy of protein synthesis. Let's say ribosome would make one mistake in 10,000 amino acids incorporated into proteins, 1 in 10,000. But we could make it even more accurate, but it doesn't happen in nature for some reason. We could make it relaxed, we could make it more erroneous. And now we have this entire continuum of opportunities. Cell could have very accurate translation, or it could have a relaxed translation to some level, to some extent. And we have all kinds of events happening over here, and some of them are very well understood right now. Let's say if mistranslation or erroneous translation is very high, the level of mistranslation is very high, it would lead to death. That's what we do with antibiotics. We add antibiotic, it causes mistranslation, and bacterial cells, bad bacteria die. And we could make also cells very, very accurate, and that's what we know much less, but we'll talk about it a bit later. So what are the benefits of altered translation accuracy? On one hand, it could be beneficial for certain situations. Let's say we have Mycobacterium. These are bacteria causing tuberculosis, many other devastating diseases, but tuberculosis is the premier case for that. And currently we treat Mycobacterium with rifampicin. So this is antibiotics that inhibits as a transcription. Transcription is writing mRNA on DNA, so writing the small recipe for the kitchen. And curiously, you can really play with translation accuracy. You can sort of make Mycobacteria translate with more errors than usual, and these bacteria would become more resistant to rifampicin. So here to the left, we see plate. It's empty Petri dish plate. This is the way we grow actually bacteria. So just to give you an idea what it is, this is a Petri dish from my lab, right? So it's a small object like 9 centimeters in diameter, and you can grow here on one plate like 1 billion cells. So 1 billion cells were plated on that agar plate, and just 2 colonies grew. So out of 1 billion, just 2 rifampicin-resistant mutants. So mutations happen. They are rare, but they happen. But here you can see many, many, many colonies. So many, so you can see almost a lawn of bacteria. So it means that mistranslation, so production of incorrect proteins, somehow allows bacteria to resist rifampicin. So it's an interesting fact for science, but it's also a dangerous observation for medicine. So bacteria will always devise a way to bypass all our attempts to kill them. And so do we. So it's an arms race. We invent something new, they evolve to resist our antibiotics. Here is another example now how mistranslation could be a problem. Now we move to animals, something more interesting. On the picture you see 2 mice. The one to the rear is called wild type or normal mice, and one to the front is so-called sticky mouse. If you look, so they are of the same age. It's 6-week-old mice. But this one is smaller, and if you look, its fur, its coat is a little bit different. It's irregular, and it looks like individual hairs will stick to each other, like it's it's not well-groomed mouse. Also, part of the hair goes away. So this mouse has problems with hair follicles. They lose hair really easily. As researchers discovered, the problem is in a protein synthesis as well. There is one gene that controls production of amino acid charged tRNAs, these small molecules that bring amino acids into the ribosome. So instead of making just one type of amino acid charged tRNA, this protein makes errors and it produces like the same anticodon but different amino acids over here. This would lead to mistranslation again, and this would lead to new neurodegeneration. So here is a wild type. This is a small slice of the follicle of the skin near the hair, and you can see here neuronal cells. So there is a body of neuronal cells, there is an axon, of neuronal cell. And in wild type, it looks pretty much like you see here. In the mutant, you see the degeneration. You don't see this regular arrangement of neuronal cells. And even those that are here are not completely healthy, as we would see on other pictures, but for the clarity and brevity of presentation, I won't show you. So what we see here that mistranslation or erroneous translation is relatively well studied. We understand that sometimes it leads to new opportunities for bacteria to resist antibiotics. We know that usually in animals, it's a problem, it's some disease. But what we don't know is how hyperaccuracy works. So it's pretty easy to isolate something which works bad, like to damage something or to make some process go wrong is not a big deal. But to make it more accurate or faster is always much more difficult. And it's even more difficult to understand why nature or life didn't want to make a protein synthesis rate a maximal one. I mean, maximally accurate or maximally fast. So we don't know why hyperaccuracy is not a physiological norm, and we don't know then what would be long-term consequences of hyperaccuracy. Let's imagine we build a cell that is maximally accurate in protein synthesis. What would happen in a long term? I mean, through many generations in evolutionary terms. So this is basically very interesting questions. And as my biological specialty comes from bacterial genetics, I was always interested to find a good model or interesting question that I could study with my bacteria of my research interest. So basically, what people know about translation and mistranslation comes from either animals or single-celled eukaryotes. So this is so-called phylogenetic tree. Biologists always like to think in terms of tree, like all life comes from single root, and then it branches off, and we have all these very different life forms on our planet coming from single source. And here is E. coli. This is another favorite object of molecular biologists. So these groups of organisms are very well studied. They are so-called model ones. But I work here. I work with actinobacteria. This is something in the middle, still very distant from E. coli, and of course very distant from humans and yeast. And it also makes interesting case to study something distant from well-studied objects, because we may stumble upon very interesting phenomena. And when we think about actinobacteria, they are interesting, first of all, because they produce a lot of antibiotics. So antibiotics, for the most part, are of natural origin. We found them in soil, in plants, and actually actinomycetes, or one specific genus within actinomycetes called Streptomyces, is one single deepest source of antibiotics. So you can see to the right a colony. So it's zoomed up colony of Streptomyces. You can see the small drops of liquid. Actually, this is exudate, which contains some pigments and antibiotics. And to the left, you see like a artistic depiction of the chromosome, the thick blue line, and chemical formulas on top, above and below the chromosome are other antibiotics which can be produced by a single strain of Streptomyces. So this is probably the most serious motive to study Streptomyces all around the globe. All researchers study in this group because we want to make more natural products for different reasons. Right now, why we need them? We need them for reasons of shortage of antibiotics in a current situation. So this happened many years ago. So actually, the boom of antibiotics was caused by Second World War. So the biggest accelerator of industrial production of antibiotics was the need to treat wounded soldiers. And you can see here the advertisement showing that how much United States cared about production of antibiotics. So big was the demand for penicillin at that time that drugstores even sold home, like do-it-yourself kits to produce penicillin in home conditions. So basically, people had to infest the bread. You got a mold on bread, and you can harvest antibiotic. Okay, so and we need them today globally because of misuse and abuse of antibiotics, and because of spread of antibiotic-resistant bacteria. We have this dangerous acronym ESCAPE, which stands for 6 most notorious and most dangerous bacterial pathogens we ever encountered. Many of them are already resistant to virtually everything that we have in the drugstore. So it's not now a question if you have money or medical insurance or not. You might be admitted to the hospital, and you might lose, let's say, a limb or something just because you are infected by one of these guys, and there is no drug which could treat them. And it could even become worse and worse if we don't come up with idea how to develop new antibiotics or how to counter these pathogens in any other imaginable way. And there is also a Ukrainian dimension to this problem because we have an active war. And so we have a flood of wounded soldiers. And many of them already harbor bacteria which are resistant to everything. You can see here a small table from a recent paper in the most renowned medical journal, Lancet. If you see R over here, it stands that bacteria is resistant to that drug. And you see it's R almost everywhere. So there is no way to treat that soldier, and he actually lost his limb. So this is a huge problem. But there's also, like any crisis, it also calls for innovation. It also reignites interest in finding new drugs, finding new ways to develop measures to counter the type of antibacterial resistance. And one of these measures is to unlock the natural potential of bacteria. They have a lot of genes, a lot of potential to produce many novel drugs. We just need to know a way how to discover, how to express it. And by playing with translation accuracy, as I will show you in a few slides, we could probably formulate a new instruments, new tools to discover novel drugs. And my talk will be divided in 2 parts over here because I will focus on two elements of protein translation machinery. I mentioned it before. I will focus first on a ribosomal protein S12. So this is a part of ribosome. This is part of accuracy check center of the ribosome. And actually, it wasn't something that was discovered by me or I was the first. Actually, the protein S12 was pinpointed already in the works of Professor Luigi Gorini, whom I mentioned a few slides above. But his work was done on E. coli, not Streptomyces. The first evidence that mutations or altered structure of protein S12 makes difference for Streptomyces came in 1996. It was done by Professor Kozo Ochi from Japan. You can see here to the left, when you see this WT, it means wild type or natural strain. It produces some blue diffusible pigment. It's called actinorhodin. And then Professor Ochi selected for the mutants harboring tiny changes in a gene encoding protein S12. So let's say we have lysine residue in this position. So remember, protein is a chain of amino acid residues, and you can sort of change these residues one to another. And so he found several mutations like K88E. So this position was changed to aspartate. This position, here double mutant, this proline, and this lysine were changed to other residues. Long story short, he isolated a large collection of mutants with carrying point changes in the protein S12. And you can see they all produce different amount of pigment, which is interesting because probably half of the efforts of current industrial microbiology is to overproduce some industrially important molecules. Just for economical reasons, it makes sense. If you can produce, let's say, 1 gram of antibiotics and pay for that $100, if you can do it for $1, it's a huge difference, right? That's why a lot of effort is invested into understanding how we can overproduce molecule. And here you can have a clear illustration. This is the wild type. It produces some amount, and here it produces much, much more. And industry is very much interested in these mutations without any interest in the mechanisms which lead from this to this. But this is kind of a short-sighted strategy. If you don't understand mechanism, you probably lose something, because by knowing mechanism, you can make it more efficiently in a more rational way. And so Professor Ochi has spent almost 30 years of his career trying to decipher the mechanism, from a single amino acid substitution to huge difference in antibiotic production. And in the end, and he retired in 2016. Before that, he visited Lviv. I had a privilege to collaborate them for 2 years, thanks to the grant from State Fund of Fundamental Research of Ukraine in that time. And he confessed that I know as much about these mutations in terms of mechanisms as I did during the first discovery. So it was a tough problem. And I was really encouraged to take on the torch of that study and probably trying to move at least one step forward in understanding of the inner mechanism that operates, that leads from a single mutation to a deep rewiring of the metabolic map of streptomyces. So this was my still a long-term research question. We want to understand this black box. So we know there is some change over here, we can describe it, we can pinpoint it, and we see the effect, but we don't know how it works. We know something, this is written on a black background, is what we already knew, but there is a lot to discover, to really feel this mechanism with all the details. And I will focus on a single position of the protein. So we have this lysine residue in position 88, and I can make a mutant which would carry either a glutamate residue instead of lysine in that position, or it would carry arginine residue instead of lysine. So very simple system. So the idea is, if we reduce the complexity of the problem to the well-studied strain, to a single event, we will probably understand more. And at that point, we have largely a descriptive picture. It's still not an insight into the mechanism, but it's a very detailed description of what we have. And on other plate, the strains do not look different. This is our wild type, so it's a parent strain. And these alones of our mutants, they look pretty much the same. But if you go deeper, if you have good scanning electron microscopy, you can see that actually they are quite different. So to the right, basically it's a wild type for the purpose of representation. This is how you can see below these chains of both. Those are spores of our strain. So it looks like typical healthy streptomyces colony. And when you go from right to left, you will see some changes. You will see you may interpret it different way, but at least we can state they differ in their morphology, although we don't really know why. This would be our next step to understand. We know that other strains do not grow differently from the wild type. Also, when we measure the dry protein mass, when we measure colony forming units. So it looks like this small mutation doesn't have any major effect on the ability of the strain to grow and accumulate protein, which is good, which would be expected. But we do see clear changes in its ability to produce antibiotics. What you see here is so-called agar-plaque assay. How it works, I'll explain in a few words. So as I showed you, you have, let's say, agar plate, right? You grow your bacterial cells over here. You can cut, and you grow, let's say, for 5 days. If the bacterium produces antibiotics, it will produce it, and it will pump it out of the cells into the medium. So it will discard it from the cells beneath into agar. And you can cut off a round cylinder out of this one, and you can stack this round cylinder on top of another plate where you seeded another bacterium. Let's say you have some bacterium that you would like to kill because it's a pathogen. So you can test different natural samples against the bacterium. You just seed it on plate, you put some drops of antibiotic or some agar plaques, and if it agar plaques kill a bacterium, you would see the inhibition of growth. So you will see it's like a transparent halo around your agar plaque. So the larger the halo is, the more efficient the antibiotic is, or the higher concentration of antibiotic is in that agar plaque. So in this way, you can test some, I would say, qualitative differences. You cannot really tell what is here, just different mixture of antibiotics or just much more of one antibiotics over another. But let's go to the center photo. You can see here agar plaque with no activity at all. So no activity. It doesn't produce under these conditions any drugs, but the mutants, these RPSL mutants, they do produce antibiotics, which is interesting as it opens a way to activate the production of new antibiotics. So remember, the current problem is that we don't have drugs to treat many dangerous infections in hospitals. And we need to find new drugs. And the problem is that big pharmaceutical companies abandoned antibiotic discovery business because it's not profitable. It's not profitable to discover and introduce new antibiotics. And it is not profitable because the return on investment is very low. You need to invest a lot, and the chances of failure are very high. And once you introduce something, the generics will follow and the price will drop, and the company will not recover the expenses they had during the development phase. And we need to become innovative. It means that big companies work often by scale. They could just do some tests on a large scale and in hopes that they will discover some valuable molecule. But we don't need a scale, we don't need a quantity, we need a quality of thinking. We need to find new ways to precisely find natural antibiotics produced or encrypted in Streptomyces genomes. And I think that it's not just my isolated idea, it's what the field thinks. Using playing with translational accuracy by changing this astral protein, we could sort of wake up sleeping genes. So there are genes which are not expressed, which don't lead to proteins, which don't lead to antibiotics then. And if we find a way to awake these genes, we might awake new chemistry. I mean, we might find new small molecules which could be converted into drugs. And this is a good illustration of this point. So wild type doesn't produce anything, but when we introduce these mutations, these all mutations wake up the production of some drug. So this is a qualitative picture. We see that there is an activation of production of some bioactive molecules to a bigger extent or to a smaller extent, but we don't know what changed inside of the cell. When we go down, we could use liquid chromatography, so we could kind of separate all the molecules produced or contained in this agar plug, and you can see these peaks, green peaks, red peaks, and we can assign a structure to these peaks using specific chemical methods. And we can see that some compounds are upregulated. I mean, some compounds are produced in more amounts, some compounds are produced in less amounts. And so we get this already quantitative picture, what's going on, but it's still very complex. Like, we don't understand how a single mutation impacts different pathways leading to these antibiotics, to these molecules. And what we tried to do this called omic approaches. When you analyze, let's say, all the metabolites in cells, or you analyze expression of all genes. And we focused on a very important messenger. It's like a hormone, but in bacterium. So when it's produced, it changes a lot of processes inside of cell. Let's say at certain point of life cycle. So here on the x-axis, you have the time, and here you have the level, the concentration of this secondary messenger. And the concentration of this secondary messenger, called cDGMP, follows a wave-like pattern. It goes up and down, and when it's up at certain time point, cells do not grow. Up, they don't grow to the air, they grow inside of agar plate. But when it goes down, they start growing up. You have this aerial mycelium, and when you go up, it branches off, it produces spores, and so on. And what we discovered is that actually you should pay attention to the graph to the left. So we have this wild type, so parent strain, no mutations. It pretty much accumulates the cDGMP throughout the course of the experiment, which is 120 hours. And one of our mutants, K88R, produces almost no cDGMP. This was a very drastic difference. The two other mutants, well, they're pretty much within the error margin of the wild type. Maybe a little bit less over here than wild type, but still not that dramatic as K88R. So this was like a big finding for us. As I told you before, my satisfaction would be even with making a one step towards understanding the mechanism. And it looks like we touched upon one of the interesting findings, which weren't known before that mutation within the ribosome somehow influences the level of accumulation of cDGMP. And this is kind of a master molecule for Streptomyces metabolism. And what's interesting is that different mutations have different effects. So all these three mutations would make a ribosome more accurate, right? So when I pick it up, type of mutations I wanted to study something which would make Streptomyces more accurate than usual during the translation. And despite the fact that all three mutations would equally affect the accuracy of translation, they have different impact on cDGMP accumulation. And again, it's very hard to explain. One more mystery that we need to resolve. We also try to understand the production of all mRNAs within the cell. We use this so-called RNA-seq technique. Basically, what we do at each given time point of a Streptomyces cell produces hundreds or maybe thousands of proteins. And we want to understand what the difference is between the wild type, the parental strain, and the mutant. How many genes or their expression is changed. The hope was to find some like a key gene which would be very, very different from the wild type. But the picture in reality is much more complicated. We see changes in hundreds of genes. Some of genes go up in their transcription as compared to the wild type. Some of them go down. And we're still trying to rationalize it. We're still trying to understand what is a primary event, what is a secondary one, which one follows the secondary event, and so on and so forth. We still have some ideas, but I would say I would think it would need much more elaboration. And finally, what we can do, we can go up to the atomic level. We can go down to the single protein as well, and we can ask the question. So how would that single mutation change the structure of the protein? So this is the domain of protein modeling. So it's completely computational work. We haven't found any dramatic changes. So we can just superpose two protein molecules. I told you that the shape of protein is very intricate and it's unique to each protein. And when you take two molecules, two protein molecules, you can sort of align them one onto other or superpose them. And you can see if there are some deviations, some distortion of these helices and sheets, one relative to another. We spotted some differences, but we think they are quite minor ones. They wouldn't explain such a gross differences in the phenotypes or in the behavior of the wild type as compared to the mutant. And current idea is that however tiny this change in the protein shape could be, it could be changes of one single hydrogen bond, probably. It might affect neighboring proteins. The ribosome, the protein synthesis machinery, consists of almost 50 proteins. And protein S12 is located between its neighbors. Maybe even a tiny change like lysine to arginine substitution would affect the neighbors, would sort of like make them move a little bit or distort their structure. And you would have this domino effect. Small change in one protein would lead to change all over the ribosome. But to find it, we need to sort of see the entire ribosome. And luckily, we have found the collaborators in Tohoku University in Japan who are masters of cryo-electron microscopy. So basically, you can freeze the ribosome almost at absolute zero. And then you can beam it with an electron rays, and the diffraction picture would tell you the structure of the ribosome. Right now, we have very crude view of Streptomyces ribosome. It greatly resembles mycobacterial ribosome, which would be expected because Mycobacterium is a cousin of Streptomyces. It's an evolutionary, very, very close relative, the same class of actinobacteria. And we hope that as we refine the structure, we would see the tiny details around the S12 proteins. And it would give us an idea how small amino acid change would have such a great effects on the metabolism of Streptomyces. So basically right now we have, as I already said, a descriptive level of work. We just have a wild type parental strain, we have some mutant strain, and we just describe them as accurate, as detailed as we can. And we see certain changes. We see changes in sporulation, we see changes in a CDGMP expression, accumulation level. But it still doesn't give us ideas. There is no this cause to effect chain of events. We still have a lot to feel between the initial event, like a mutation in a gene for S12 protein, to increased, let's say, antibiotic production. But I think we have a very good model. And we have good collaborators right now. And already we have, even at the descriptive level, we have interesting findings of connection between ribosomal mutation and CDGMP accumulation. And this molecule comes directly from GTP, which is a central molecule for like an energy source for protein synthesis. So probably there is a nexus point where these both processes converge and interact and impact either unidirectionally or bidirectionally. This is what we currently pursuing. Also, what's worth noting is that many of the effects that we are studying, like antibiotic production, they happen in old cultures. Even when we compare it to human life, the antibiotic is produced by bacteria in the retirement age. It's not young culture, it's old culture. And so the most relevant effects of repair cell mutations are those happening in cultures that are almost at the death stage. And this is actually barely studied part of bacterial physiology. Almost everyone likes to study here. So it's a young culture, young and beautiful, it grows, it's active, everything's good, it's easy to study. But here it's very difficult because many proteins get degraded, mRNA degraded, cells senesced. So, but for our specific case, this is where actually we are with antibiotic production. And by making this mutation, probably we kind of prolong this productive phase. Would it make sense? We don't know. We actually went from the organism and we went down to the model of the protein, and we will fill up this entire space of opportunities to understand the mechanism. But there is one interesting work which shows that actually it could make really sense. It was published in 2021. So basically, the researchers isolated mutant mouse, which not only mouse, but also yeast, worm, and fly, which had a mutation with protein, which is relative. It's the same, but different name. It's S12 protein, which makes this organism live longer. So probably by manipulating ribosome, by manipulating the accuracy of translation, we could prolong the life expectation of streptomyces culture, but maybe also of animals. And intuitively, this would be expected because when you think how the ribosome could be more accurate. So it's like building a chain. You add one element to another, and you can do it faster or you can do it slower. And faster means with more errors because you do it faster. So you pay less attention what you stick in to each to other. But when you do it slower, you have more time to check what you're doing. And in that way, your protein synthesis rate would slow down, but your life span would enlarge. Would it work for animals? We don't know, but this is very provoking paper, which shows that probably playing with translation accuracy is interesting not only for antibiotic field, but also for making human life longer. Okay, now let's switch the gears to second and the last part of my talk. So we focused so far on the protein S12, which is part of the ribosome. Now we will focus on the tRNA. So this is my component which brings amino acid into ribosome. So these are the logistic partner which taxis amino acid residues inside of the protein synthesis factor. Why is it interesting? Well, because it's highly modified molecule. So tRNA is a 78, 76 nucleotide long chain, which is folded into clover-like structure. This is how it's depicted in the textbooks. Actually, it's a little bit different, but it's good enough. And each of these nucleotides is somehow modified. Some small chemical groups are added or removed or excised or substituted, and it's all needed for the functional reasons. These modifications make this molecule functional, so it could accept the amino acid and it could bring it into the ribosome and decode it. And decoding means interaction between the anticodon of the tRNA, which is at the bottom of our clover leaf. So these are positions 34, 35, 36. These three, the most important positions of our tRNA, they interact with our instruction. So this is how ribosome knows that it needs to include into protein certain amino acid. We will focus on, as an example, on it's the same but shown in a more schematic way. So we will focus only on this anticodon loop, and these three nucleotides are very important. If you have here AU, it will interact only with UUA on the messenger RNA. So this is like a rule how it works. And you need certain modifications on this position and on the neighboring positions, and we will focus on this positions called 37. And this red dot on over here, it depicts certain level of modification. So basically, cells produce proteins which would modify this A to make molecule exist. So you will have thiomethyl group over here, you will have isopentenyl, hydroxylated isopentenyl over here. So the natural A looks like that, and the hypermodified A, or adenosine, looks like that. And we will depict it further like this. So three different figures stand for three modifications of this nucleotide. So our question was, if we could play with translation accuracy by changing S12 protein, we probably could change translation accuracy by changing tRNA. If this position is important for accurate decoding of UUA, by erasing this modification, we probably could make translation more relaxed. So we would have some mistranslations, some errors in protein synthesis, or maybe not. We didn't know exactly, because this question is actually a little bit controversial in scientific literature right now. Well, basically, we built such a mutant. We built two mutants which could not produce these modifications. So here is the wild type. So it has all the modifications, both triangles, quadrangle, and circle over here. You have healthy, good-looking spore chains of streptomyces. But when you have a strand which is not able to put on this modification onto position 37, you have what we call a sick culture. So it looks like sick. It's not healthy. It has no spores, and the mycelium looks a little bit distorted. And when you look on plate, it looks different. This is wild type. It's a whitish yellow because of production of spores. This culture doesn't produce any spores. So basically what we see here, again, it's a description that by screwing up the tRNA modification, we screw up morphogenesis. So there is a connection between tRNA modification and ability of strain to produce spores. Okay, we are always interested in antibiotic production. This is our main motivation, to find new ways to discover novel molecules. And here again, we see differences. So wild type is on top, and you can see that one of the mutants become a little bit reddish. It starts producing red pigment called polymycin. We know its structure. For some reason, this pathway is turned on upon loss of gene involved in modification of tRNA. Again, we see difference in antibiotic production. So the wild type produces some antibiotic. You can see this inhibition zone around the agar plaque. And the mutant, no antibiotic production. And this test culture, this white dots all on the background are bacterial cells that can grow closely to the agar plaque of the mutant. So no antibiotic production. So here you have, as a test culture, we used Bacillus cereus. So this is a bacterial cell. We can use also eukaryotic cells. We used a yeast, the Baryomyces hansenii. And again, and here you can see opposite picture. The wild type, almost no production of something that could kill fungi. So these fungi grow close to the agar plaque. And here you can see this transparent zone because our mutant can produce some antibiotics that kill fungi. So I would say this is the main problem of studying these mutations. You don't see well and pronounced effects in one direction. Like we would expect that single mutation would lead to either activation of all antibiotics or probably canceling out its production, but it's not the case. Some genes go up in their transcription, some genes go down in their expression. And it's really hard to rationalize how single mutations would lead to such an opposite effects. Well, I mean, we need more experiments to understand it. We also saw differences on the level of susceptibility to hydrogen peroxide. The hydrogen peroxide is like a test molecule to understand if cell is able to withstand oxidative stress. And the result was kind of counterintuitive. We would expect that our mutant would be more susceptible to hydrogen peroxide because it would mistranslate proteins. It would not be able to produce right protein to protect itself from hydrogen peroxide. But actually, no, it's more resistant to hydrogen peroxide than the wild type. And finally, and the most interesting, I would say, effect, which probably is what we would like to understand most of all, is it was also discovered by accident. It was like a mistake in the experiment of one of my students when he plated wrong strain on the plate. So he mixed up the strains and antibiotics, but we forgot about these plates for 1 week. But when we took them out from the incubator, we observed a really strange picture. So we have a very strong antibiotic called novobiocin. And here is a paper disc impregnated into novobiocin. So this antibiotic diffuses from the paper disc all around. And if concentration is strong enough, it would kill streptomyces, it would not grow. So you actually don't see the growth. And this white stuff around, this is the growth. So actually novobiocin very effectively kills streptomyces at certain concentration. But then at certain concentration, it stops being effective. That's understood. So this is expected picture. The same we observed for one of our mutants. This mutant would not lose all the modification, not all three, but just one of the modification on our tRNA. But when we have a strain completely devoid of modification on position 37, we observed this result. You probably see these small round colonies around the novobiocin disc. So there is no clear and clean zone of growth inhibition. Within the zone of inhibition, you can see these small colonies. So these colonies are somehow resistant to novobiocin. So the classical idea of bacterial genetics is that these are mutants. So they just spontaneously mutated, and some of these mutations are beneficial. They help these bacteria to grow in presence of novobiocin. But the counter argument is that there are too many such colonies around this disk. This mutation should be very rare. We would expect one probably colony, and maybe not at all colonies. But you can see here tens of colonies. And I zoomed in on this effect again. So you can see these colonies. We picked up these colonies, and again, we tested their susceptibility to novobiocin. They are still susceptible. They don't grow completely to the edge of the disc with novobiocin. They stop growing over here. Well, some of them do grow, but some of them didn't. So it's probably tells us it's not a mutation, it's called a persistence. So cells of streptomyces are able to switch on some mechanism to go into a deep sleep for some time until antibiotic decomposes on the plate, and then they regrow. This was described for several pathogenic bacteria. And the persistent phenomenon is very interesting and is currently heavily studied on different models. What's interesting in our case is that it looks like we are able to induce the persistence with the help of mutations in tRNA modification genes. This wasn't described before. And again, we have a description, we have the effect, we don't understand the mechanism. And well, for me, it's probably the biggest reward in science to decipher in this puzzle. So we basically brainstorming all possible ways to study this phenomenon. Besides basic interest, it's of course very interesting for antimicrobial resistance. So I heavily focused my talk on the urgent need for novel antibiotics. It's indeed true. We need to discover novel molecules. We need to discover novel drugs to prevent the spread of antimicrobial resistance, to treat the infections. But we also need to understand the mechanism of antimicrobial resistance. When we understand them, we can prevent them. And it looks like we still have much to learn how bacteria become resistant. And it doesn't matter what the objects you are studying. You can study streptomyces, it will be relevant for staphylococcus, for enterococcus, for all the pathogens. So we're really happy. Well, we're not happy that persistence exists because it's a source of problem for healthcare. But it's interesting that we found one more point of entry into the persistence state. And what makes us believe that it is persistence and not mutations, we observe it on different antibiotics that work through very, very different mechanisms. So it means it's not something like mutations. It's more like an ability of our mutant to become a persistent, and somehow this ability has been unlocked upon the mutations, upon the loss of the modification on tRNA. We still don't know if it's related to mistranslation. We still don't know if it's related to translation accuracy, but anyway, tRNA is involved only in translation. So there is no other way it would affect the bacterial physiology, this way or another. So currently, with our tRNA studies, with modification, we are at the level of descriptive science. So we describe some phenotypes, we verify and re-verify them to make sure that it's true, or it's not some artifact. And we are pretty much sure that we have really interesting observations worth further studying, and we're building new collaborations to understand this. Because our current picture carries a lot of interrogation signs, which means this is what we don't understand. And we eagerly want to understand for many reasons, both the practical ones and fundamental ones. And I wish, first of all, thank to all my co-workers in Lviv University and my collaborators, most prominently Professor Koza Ochi. So it was back a decade ago when it was safe to travel to Ukraine, and he visited us. And it was a big opportunity and inspiration for all of us. And right now, our work is funded by a large network of grants, and the core ones are described here. And I would single out the funding from German Ministry, BMBF. But throughout the career, I had an opportunity and privilege to be funded by many agencies. But money are important, of course. I started with them. But it's even more important to have good teams and good collaborators. And here they are. There are many. I wouldn't list all of them. I just list a head of groups, head of the laboratories whom I collaborated with. And finally, I thank you. I will be happy to take your questions.
Thank you very much. Thank you for introducing us to a absolutely interesting and fascinating topic. While our fellows are thinking of a question, I would ask you maybe two questions more related to how do you do your research? First of all, I understand that the quality or specifics of equipment probably plays a lot of role in your research, and how do you overcome this obstacle?
Thanks, Victoria. You're right. This wouldn't be impossible without all the people and their willingness to collaborate. Of course, you can see it in Japan, Germany. I'm trying to find just top collaborators. But I always be able to find them because I think you just need to pose interesting question. If you have interesting question, you will find it. And not trying to be like arrogant, but it looks like the question is interesting one. That's why I wouldn't do even 1% of that without the input of my German, Japanese, and Swedish and American partners. But still, the idea is the driver of science. Like one needs to have a good idea. I mean, it shouldn't be even brilliant because I don't consider myself brilliant. It should be good enough. And it's good in science. It shouldn't be something outstanding, just good enough workable hypothesis. And despite really low funding, I would say in Ukraine, it steadily goes up. I wish it could go faster. I wish we could have more funding, but I already hear from returning back from Harvard, it's already 17 years. And over the 70 years, I could tell you that it's getting better, even taking into account an inflation. So I would say Ukraine, the government is getting more serious about science. And the seriousness is reflected in the amount of grants. And we also have all the opportunities of the world. Like now, world is heavily interconnected. There is no, voice. You can contact everyone by email and ask, hey, can we do that or this? And that's it. So the short answer would be just knock the doors and you will find the equipment.